Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by AGTC, Allergan and Abbvie Company, and Novartis. We'd also like to thank Accessible Media Inc., our national media partner. Everyone here should download the AMI app, which is a fabulous resource. And you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below.
And welcome everyone. We're really excited to have, as Morgan mentioned, two experts to talk about two different aspects of glaucoma. We'll talk to Dr. Chauhan first, who is a researcher who's doing some really exciting new research that FBC is so proud to fund. And then we'll talk to Dr. Kanjee about drops and medicines and other things about glaucoma. So welcome, Dr. Chauhan. Dr. Chauhan is the Mathers Professor and Research Director of Ophthalmology and Visual Sciences and Professor of Physiology and Biophysics at Dalhousie University. And he'll be talking about his project that FBC has just announced that we're funding starting in 2022. And his broader research interests are on changes in the visual field and the optic nerve in glaucoma. So we'll get into some of that next. So yeah, so welcome. Thank you for joining us.
Thank you for having me, Larissa.
So some of us may know, and for people in the audience who don't know, in glaucoma, there is high pressure in the eye, and this can lead to damage to a part of the eye called the optic nerve, and in particular to types of cells called retinal ganglion cells. I was wondering if you could tell us a little bit more about retinal ganglion cells and the role that they play in vision loss caused by glaucoma.
All right. Okay, so first of all, thank you for having me and giving me the opportunity to talk about this research. So as you rightly said, glaucoma is caused typically by high eye pressure, intraocular pressure, but not always. So we're not entirely sure whether it's the pressure that's the sole culprit of damage. But one thing that is for sure in this disease, and that is the defining feature of glaucoma, and that is these cells that you've mentioned, retinal ganglion cells. So they're specialized neurons. Neurons means it's a type of cell that resides in the brain or the central nervous system. And this type of cell is actually very special because it carries visual messages from the eye to the brain. So you can imagine that the little cell body lives inside the retina, which is in the eye, and it has essentially an axon, which is like a little wire, if you like, that connects the eye to the brain. And what happens in glaucoma is that this cell dies. And what we want to do, of course, is try to understand why this particular cell type dies. What is the environment around it that's toxic, that makes it damaged and eventually die and cause visual disability, and in some cases, unfortunately, blindness as well. So in the human eye, we have between 1 and 1.5 million of these cells. And we lose some of these actually just by normal aging. But what happens in glaucoma, unfortunately, is that the rate of loss of these cells is much, much greater. And so what we try to do, both in the lab and in the clinic, is to slow down the rate of loss of these ganglion cells. So essentially, they are very specialized cells that carry visual messages from the eye to the brain.
That was a really great explanation. I always sort of think of the retinal ganglion cells almost like an electrical wire,
exactly.
Joining the retina to the brain, this like really long series of cells. I didn't realize there were so many cells that made up the optic nerve, actually.
Yeah.
You mentioned that not all types of glaucoma might be caused by pressure. Can you elaborate on that a bit? What other things might cause glaucoma?
So actually, we don't know if all types of glaucoma are caused by high pressure or not by high pressure. And there's a very strange paradox in glaucoma, and that is that about 90% of individuals, or maybe even more, of individuals who have statistically high eye pressures actually don't have glaucoma, or in fact will never get glaucoma in their lifetimes. They are at higher risk, but it doesn't necessarily mean that more of these individuals will develop glaucoma. But on the other hand, around 50% of patients with glaucoma actually have pressures that are statistically normal. So eye pressure itself doesn't distinguish terribly well between patients who have glaucoma or don't have glaucoma. But having said that, an eye pressure of a given value in my eye might be very dangerous compared to that same eye pressure in your eye. So we can't rule out the influence of eye pressure itself, irrespective of what the level is, right? And that's because, for all types of glaucoma, the only currently acknowledged treatment is actually reduction of that eye pressure, even if that eye pressure is within normal limits. So, some of your listeners here may have heard the term normal pressure glaucoma or normal tension glaucoma, and they may wonder, well, if my pressure is normal, why is my eye doctor want my pressure to be even lower? Because we know there's now very good evidence that even lowering pressure in eyes that have statistically normal pressure actually is beneficial. So there is evidence that pressure, no matter what level it's at, is actually dangerous for the eye. And that's the reason why we have this large spectrum of damage across the different range of intraocular pressure.
And I think that makes sense, the idea of it being relative. So what is sort of considered normal for one person might not be for another person. Precisely. And that, to me, suggests that it's really important to sort of go for regular eye exams, because then your doctor might be able to see an increase also, like relative to yourself, as opposed to just a one-off measurement. Right.
But remember that the diagnosis of glaucoma is not made on the basis of the eye pressure. It's made on the basis of the appearance of your optic nerve, of your retina, and of course of your visual field, right? The eye pressure is corroborative, but it's not definitive.
It's more of a red flag.
It is a red flag, but it's something that obviously eye doctors have to monitor. But the diagnosis of glaucoma is not based on the intraocular pressure measurement.
Okay, I am learning a lot, because I think I had assumed that a lot of it was based on the eye pressure. So thank you. I know we got a bit off track actually from your research, but I thought that was really interesting to learn. Thank you. So sort of getting back to what you were talking about the retinal ganglion cells and wanting to know more about how the retinal ganglion cells both look and how they're affected in the eye. So can you explain a little bit about what your project is and what challenge your project is trying to solve?
Right. So basically, we have a two-pronged approach in this project. The first part of it tries to explore the cellular mechanisms of how these ganglion cells die. And what we want to try to do is to eventually do this in humans. It won't be tomorrow, it may not be in 5 years, may not even be in 10 years, but we want to try to understand signatures that we can measure with imaging. So as you know, imaging is exploding in every field of medicine, and ophthalmology is no exception. It's remarkable, in fact, what we can see in the retina. So we are now able to actually see individual ganglion cells in the eye, intact eye. So in other words, for example, so this project, we're doing these experiments in mice, and we're able to image these individual retinal ganglion cells in mice. And we're looking for signatures of how these ganglion cells change when we create a glaucoma in these mice. So we're tracking these individual cells over time. And then what we want to try to do is to see what these changes that we see in the live animal actually represent as far as the cell health is concerned. The idea being that by looking at these different signatures that we see with our imaging, we can say, well, this cell is in danger, or this cell might be able to be rescued, right? Because it's in trouble. So we're actually trying to correlate what we see biologically under the microscope with what we can see in the intact retina in the eye. So it's very exciting because there is a field of medicine called transgenic medicine. And if you don't mind, I'll just take a couple of minutes to explain this to your listeners. So one of the beauties of this type is that we can actually engineer mice to express certain types of proteins in the eye. So for example, For this particular type of mouse, we can get it to fluoresce. The retinal ganglion cells can fluoresce, so we can actually watch the changes in the fluorescence. And this allows us to actually look at a cell as if we were looking at it in a piece of tissue under the microscope. So we can do this in a live animal. So that's the beauty of this approach. So we can measure these different signatures. Now, the second part of the project is that we are going to try to introduce a compound into the retina that can try to rescue these ganglion cells to stop them from dying. And we will do this with a technique which is called gene therapy. So what we try to do is we introduce a gene into the retinal ganglion cells to actually try to reverse the process. So that's the second part of the project. So it's very ambitious, but the two parts are quite related to each other, because the second part will tell us, because obviously sometimes we will not be able to rescue the cells that we want to rescue, but we may be able to find signatures from the first part of the project that will tell us what the opportune time window is for this rescue. So that's the project in a nutshell.
Thank you. Just actually before we move on, I think something is brushing if you've got a mic.
Oh, right. So, okay. Yeah. I'll just hold it up. Sorry.
Perfect. Thank you. So that was a lot of really interesting information. So I'm going to try to unpack it a little bit. So First of all, you've been mentioning the word signatures. So can you explain what exactly you mean when you say a signature?
So neurons, these types of special cells, they have a cell body, which basically does all the hard work. And then essentially you have what are called dendrites. And dendrites are little arms, essentially, that link one cell to the others. Now, one retinal ganglion cell can talk to hundreds, maybe thousands of other retinal ganglion cells. And what we see typically when a cell starts to show damage is that these dendrites start to retract. In other words, they pull their arms in because they're in trouble, because they are losing connections with the other cells. And this is one of the very first processes of the cell death. Now, we can image the entire retinal ganglion cell in these specific transgenic animals. So we're able to track them very, very carefully and very easily. So that's, for example, one signature that we can see. Another signature is in the change in fluorescence, because these cells fluoresce, and the changes in fluorescence can tell us something about the cell health. So these are the various things that we can actually track in the live animal. And this is what we're going to try to do. You're muted, Larissa.
Thank you. Somebody was saying there was an echo. I was going to mute in between to see if that helps. So what is the benefit of doing this work in, say, live animals versus under a microscope? Because it feels like that's like a really important part of this project.
That's a great question, Larissa. The reason we want to do this is because we can track individual cells as the disease progresses. One of the problems, of course, when you do studies under a microscope is you only have a snapshot view of what's going on. The beauty of this is we can actually track these animals in the same way that we track patients over time to see how their disease is changing. And we can do exactly the same. And that's the strength of this technique, is that we can track individual retinal ganglion cells in the eye. So in fact, this is a very powerful technique that's been developed over the last 10 years.
I don't want to get into too much technicality, but I know that the whole basis of the project is the really sort of exciting technical aspect of the imaging. So I was wondering if you could, if it's possible, in a sort of brief, high-level way, tell us what is so advanced about this new imaging technique.
Sure. So we have a specialized camera, which is actually based upon the sort of cameras that many of your patients are being imaged with. The difference is that we have a very good collaboration with a German company that makes these devices for us. And all the optics have been changed so we can actually work in the mouse eye. The mouse eye, I'm sure most of your listeners have seen mice before. Mice are extremely small. And if you look at the eye of a mouse, that's even smaller. It's remarkable how small it is, yet our device has been adapted so that we can actually do this beautiful imaging in mouse eyes. So we have to be able to get the light into the eye through a pupil, which is the hole essentially in the front of the eye that allows light to pass through. of pupil that's maybe at the most a millimeter in diameter, and we're able to image through that. So it's a highly specialized piece of equipment. We have very specialized lasers, and we have very special filters in there that allow us to do this type of imaging, plus a lot of very, very sophisticated software that allow us to track these individual cells. So we're very happy to have this device with us.
And in terms of, you mentioned gene therapy, that the second part of your project
Yes.
will be testing if a gene therapy approach
Yes.
could, I guess, stop the retinal ganglion cells
Right.
from dying. Do
Right.
you want to explain a little bit more about what sort of gene therapy that would
Right.
be?
So we're using, like the approach that's been used in some retinal diseases. So we use a virus-based approach, which is an adeno-associated virus. And these viruses are quite safe in the eye. And essentially what you do is you take this virus and you engineer the virus to be able to seek out a specific cell. So what you can do is you can actually pack it with a type of sensor, if you like, which will latch onto a specific type of cell. So we can make sure that when we inject this into the eye, the virus goes specifically to this particular type of cell because its cell membrane expresses a certain protein. So we can tag the virus to essentially hit that particular cell. And then we can get the virus to go inside the cell and essentially deliver either receptors, or it can deliver, in our case, something which we call a brain-derived neurotropic factor. So this is essentially a nutrient that the ganglion cell needs in order for it to survive and to do well. So this particular gene therapy approach is going to deliver this compound to the retinal ganglion cells and actually also get it to increase the expression of the receptor so that it binds, so that this neurotrophic factor binds to this receptor so that it can be, the cell can be made more functional. So this is the general idea of how this particular gene therapy approach works. It's been used now in other models as well, preclinical models. And the great thing is that this particular approach of gene therapy is used in actually many retinal diseases and optic nerve diseases as well, with actually very good success. So it's not science fiction to think that one day this will work in glaucoma. So we're very excited about that.
It is really exciting. So you think that this potential gene therapy, do you think it could make it to the clinic at some point, or is this more of a proof of principle?
Yeah, so exactly right. So our project is a little bit mechanistic. In other words, we want to try to understand the processes about how it works. And I think once we understand that, we can tailor the therapy, the gene therapy, to be much more precise. Of course, one can do this in humans right now, and I think it's relatively safe. And in fact, some laboratories in the world are doing this approach. That is one way to do it, but we're a little bit guarded in the sense that we want to try to understand the mechanisms, to understand, if we have any unwanted side effects that are not foreseen in human studies. And it'll probably allow us to tailor the types of titers, the doses that we need in order to get the optimal effects. So our project is a little bit mechanistic, but I think eventually this will help into the translation of this going into the clinic. So, from a time span perspective, as I said, this is a long-term project. I don't see this happening in the next 5 years or 10 years, but I think we'll make great strides towards this being an additional therapy in glaucoma, besides the reduction of eye pressure, which will always remain the most important approach to treating the disease.
Great. And I know with the eye pressure drops, we'll be getting to Dr. Kanjee soon, who will be talking a little bit more about that. Right. So I have sort of two follow-up questions just about the specifics of the research. The first is, this new technique you're talking about, or imaging technique, allows you to image the retinal ganglion cells in currently in mice. Do you think this would be a technique that could be used to diagnose or look at the progression of glaucoma in humans, or is it very much in the laboratory?
This is also an objective for us to be able to introduce this diagnostic technique for humans. There are a lot of technical challenges that we have when we try to do this, but ultimately, yes, this is the plan. I have another portion of my laboratory that's trying to do this type of work to see how we can translate this into humans. It's difficult because there are a lot of safety issues yet that we have to consider. But for sure, this would add a tremendous amount to our current diagnostic capabilities in glaucoma if we were able to do this. Because even though the tools that we have, that your patients may have visual field tests done, they may have imaging techniques done, which are much, much better than what we had even 10 years ago, they're still relatively crude. They don't tell us about individual cells. It's a very gross measurement. It's a good measurement, but it's still not as precise as we want it to be. So one of the objectives of this type of work will be to translate this into humans so that we can actually do a quantitative measurement of how many ganglion cells are still remaining in a particular eye, which I think would be a phenomenal advance in our field.
Yeah, definitely, just to get sort of a richer amount of data, give you a more precise understanding of how it's progressing. So maybe you've already talked about this, but is there anything else that you want to add in terms of sort of the longer term impacts that this project could have? I know you mentioned already the gene therapy as well as translating this into humans. Is there any other sort of aspects that you feel like, if this project is successful, could be useful in the future?
Yeah, I mean, I think it would open up a lot of other avenues. So, for example, if we can show that delivery of this approach, of this virus-based gene therapy is successful, it's quite possible that we could engineer a virus to have other compounds in it. So, for example, to target different parts of the cell cycle, for example, that we can rescue. We may be able to tailor different types of drugs, for example, depending on the age and the stage of the patient in terms of damage. There are a lot of potential things that we can do. So our objective in this project is to just show that we're able to image this, that we're able to track this over time, and we're able to show success. And I think that once we're able to do that, there are a whole lot of possibilities to be able to translate this work.
Great. So just as a final question before we bring Dr. Kanjee here, I know you've got a very large research program, and this is just one part of it. I was wondering if you wanted to tell us a little bit more about sort of your larger scope of research and some other exciting projects that you have going on in the lab.
Yeah, so I spend my time, actually I spend most of my time doing clinical research, which is doing research with patient data as opposed to being in the lab. But this particular grant is for my lab research, which I'm also very keen on. So I have a very large program on trying to quantify how this disease progresses in human glaucoma. I'm particularly interested in looking at the interaction of age with glaucoma. So, age is unfortunately, we're all aging daily, but age exacerbates the impact of a lot of diseases. And I think we've underestimated how much that is. So we're trying to understand how to actually to see how individuals who don't have glaucoma change over time. And actually they do change substantially. And how different these patterns are in glaucoma and why they're different in certain patients. So we're trying to establish what we call phenotypes of progression. Some glaucoma patients, once they're treated, they actually never progress. They fortunately remain stable, and they have functioning vision, actually more than functioning vision. They're able to do everything that they want for the rest of their days. Unfortunately, some patients progress very fast. And we don't really understand why fully that is. We've ruled out things like intraocular pressure, we've ruled out family history, but there's something about certain types of eyes, certain types of optic nerve characteristics that make those patients progress much faster. So a lot of my research is trying to uncover what it is that makes these patients progress faster. And the whole clinical program is really devoted towards looking at how the disease changes and how we can best detect it. And my clinical side is, of course, trying to establish these models of glaucoma, how to quantify this loss. And what I try to do with both avenues of research is to sort of have both of them talk to each other. So in other words, is my clinical research relevant to my basic science research and vice versa? So that's generally the research in a nutshell.
That's really wonderful. Actually, I can just see as you're talking about it, how a really technical aspect of this project we're funding, how it could directly impact also both the clinical research that you're doing,
Right.
but also clinical diagnosis. It's
Right.
really nice to see that big picture. Thank you so much for talking to us. And I know you're going to stay
For
sort
sure.
of on the line. So if people have questions, once we get to the Q&A, you'll be here to answer them.
For sure. Thank you, Larissa.
Thank you.
All right. Dr. Kanjee, you're up. Welcome back, and thanks for joining us again. We had a really fantastic session with you last year where we talked a lot about glaucoma, sort of what it is and how it's treated and what some of the new sort of research is happening. But today we were hoping we could really kind of focus in on drops and treatments, because we get so many questions from people about eye drops. So could we maybe just start off and talk a little bit about how do eye drops help someone who has glaucoma?
Sure, and thank you so much again for the invitation, Morgan. I really do appreciate the being able to be here. And of course, greetings from sunny Winnipeg, where we're expecting 30 to 50 centimeters of snow in the next 24 to 48 hours. It's great. So we'll talk a little bit about eye drops this afternoon. So I guess your question was, how do eye drops help people with glaucoma? It's sort of the very top level overview to this is, at the end of the day, the most important role that eye drops have is to help bring down that eye pressure, the intraocular pressure that Dr. Chauhan spoke about a little bit earlier. And so we know in glaucoma that any degree of eye pressure that the nerve is unable to handle is pressure that we want to avoid. And so by putting somebody on eye drops, we can help bring that pressure down through a number of different mechanisms. And so that would be the one common theme that binds all of the many different types of eye drops together. There are some of the newer drops that are out there now that have theoretical benefits to protecting nerve tissue over time. That's been demonstrated in certain experiments, but they're still fairly early in terms of their use in day-to-day clinical practice. So it'll probably be 10, 15 years before we know for sure whether those particular drops have that touted benefit. But even if they don't, they still help with the eye pressure, which we know is the primary driving benefit for all eye drops.
Okay, so you've just mentioned this, that there's a lot of different kinds of drops. So are there different categories of drops, or what makes these drops different from one another? And as a physician, how do you decide what drops that you prescribe for someone?
Yeah, so those are some great questions, very practical questions. The way that I like to think about it sort of in big categories is we've got drops that either reduce the production of fluid in the eye or drops that help move the fluid out of the eye a little bit more. So you can almost think about it like plumbing. And so those are sort of our broad categories. And the ones that reduce the amount of fluid being produced are probably ones that we use a little bit more commonly. But some of the drops actually do a little bit of both. So they both reduce the production and increased the outflow all at the same time. And then in terms of how do we choose which drops to use, well, I also like to talk to patients about the fact that a lot of our drops, we will consider them to be long-acting drops. And in other drops, we may say, you know, these are actually short-acting drops. And I like that distinction because that helps people understand that the long-acting drops, you probably don't need to be using them more than once a day, whereas the short-acting drops, you have to use much more frequently. When we're sitting down with the patient in front of us, we're often going to be thinking to ourselves, how can we lower their eye pressure in a way that is going to be overall safe in the long run to help with their glaucoma, but it's also going to be safe in that it's not going to create a whole bunch of additional side effects from the drops, either for the eye or for the rest of the body. And we also want to minimize the impact that we have on their day-to-day lives. Because you could be putting a drop in every couple of hours to try and keep the pressure down, but that's not very practical. So typically I'm going to begin by trying to prescribe a drop that is as infrequently used as possible. So once a day is ideal, and that's not going to have any effects on the rest of the body if at all possible. And that would sort of what I consider be my first line treatment. And then as we need to get the pressure lower and lower, we start adding more of these drops. Often we start adding the short-acting drops a little bit later on. Because they're short-acting, they have to be dosed multiple times a day.
So you mentioned, that you're trying to reduce sort of side effects on the person, both in the eye and overall. So what are some of the side effects that people might experience from their drops, or are there common problems that people have with eye drops?
Mm-hmm. So there are many, many different side effects that people can have. I guess it's important to acknowledge that most people actually don't have very many side effects, or just minimal side effects. So most people do just fine with whichever drops they're on. Common things in terms of the eye itself would be having some irritation shortly after the drop goes in, or stinging from the drop. Those are not uncommon at all. Some people develop redness or dryness to the eye, where when we're looking at these individuals under our microscopes in the clinic, we can see little dry spots on the eye, and we know, okay, people are going to be a little bit uncomfortable with those. And that usually is quite amenable to treatment with just an artificial lubricating eye drop that you can get at any pharmacy. So those would be fairly common things. Some of the drops have very, very specific types of side effects, and those are different between the different classes of eye drops. So not every eye drop is made the same way. It doesn't have the same medication in it, and each medication may have its own series of side effects. Some of them are not all that bothersome. Others can be quite bothersome, and your physician will really tailor the treatment based on your own eyes. And then occasionally the drops do have effects to the rest of the body. And so there are certain situations where we may not use a particular drop. So for instance, a very commonly used drop is one that we call a beta blocker. This is a type of medication that has been in use in medicine for decades and decades and decades, and we know it very, very well. It's used often for the management of blood pressure. But in this particular case, it happens to be used for eye pressure. But we do know that this type of medication can make people's asthma worse if they happen to have pre-existing asthma. It causes problems with the lungs and with breathing. And so someone who comes into my office and I'm thinking about what medication to start them, I'll take a look at what their medical history shows. And if I find that they have asthma and they have to use puffers several times a day, I'm probably not going to be putting them on this particular drop, or at least I'm going to be checking in with their family doctor or their respirologist or their lung doctor to make sure that they're safe to have this type of medication. So that's just one example in the host of all of the different things that we may consider.
So interesting, and this is probably very silly on my part, but I think about putting an eye drop. I don't necessarily think about that eye drop having a systemic effect in your whole body, but it's a medicine and it's going into your body. So of course it could interact with other drugs that you're taking or other conditions as well.
People don't really realize that the eyes are connected to the throat. So there's little tear ducts right in the corner by where your nose is, and that's where your regular tears drain out from. Those actually drain into the nose, sort of towards the back of the nose, and that drains into your throat, and that obviously ends up in your stomach. So any of the folks in the audience who take eye drops themselves may have had the experience in the past, if they take a particular eye drop, of thinking to themselves, oh, I've got a bit of a funny metallic taste in my mouth. What is going on? I just put an eye drop in. But oftentimes that can actually be a little bit of the drop getting down the throat, and you taste that. And that can actually, as we mentioned, end up having an effect to the rest of the body, or a systemic effect, as we might call it.
Oh yeah. So is there a proper way, while we're talking about administering the drops, is there like a particular way that people should administer the drop to their eye?
So I like to look at this from a practical point of view. And the bottom line is, if you're getting the drop on the eye, you're doing okay. So as long as the drop touches the surface of the eye and it kind of spreads over, in a single drop of medicine for glaucoma, there is more medication in it than the eye would need in that single drop, because the people who made this, they know that you're going to get the drop in, half of it's going to end up on your cheek, and that's okay. As long as the drop just kind of touches the surface of the eye, it'll immediately spread over the eye, it'll coat the eye, and there'll be enough medication in there to be absorbed to perform the effect that it needs. So that's kind of the bottom line. So if people are worried, am I getting enough drop in, half of it ended up on my face instead, as long as the drop made contact with the eye, that's okay. And sort of the backup to that is, if you're not sure, it's probably okay to put a second drop in and make sure that it does hit the eye, and then you're going to be okay. And then in terms of how do you get the drop in, well, there's lots of different methods that people have described. So often people will talk about taking a bottle and holding it above your eye and squeezing the bottle and letting the drop fall in, maybe pulling down on your eyelid to create a little pocket there, and that works very, very well. Some people do have a little bit of an issue in terms of dexterity, in terms of being able to hold a bottle up or squeeze the bottle. Some bottles are a little bit more stiff than other bottles, and so they're more difficult to squeeze to get the drop out of. And so they may find that just aiming the bottle is a little bit more challenging. So what I'll often recommend to people is find a family member or a friend who's happy enough to come and put the drop in for you, because then they can actually watch the drop go in. Or if you're not sure yourself, a method that I'll often recommend to my patients is you lie yourself down on a couch or on a bed, or even just over the side of a chair. You balance the bottle on the bridge of your nose. So you've got the dropper pointing in the direction of the eye. And so now you're flat, the bottle is on your nose, you squeeze the bottle and you kind of tilt your head towards the direction of your eye. So if you're trying to get the bottle or the drop into your right eye, then you would tilt your head a little bit to the right. So your eye is now pointing a little bit down. Then as you squeeze the bottle, that drop is going to fall right in the corner of your eye, right by the nose, and it's just going to immediately wash itself into your eye. So that can be, it's a bit more of an involved process, but if you're not sure, that's one way of doing it.
That is a fantastic tip. And I don't know if there's other parents out there who can relate, but my daughter had pink eye when she was 4, and getting those drops in her eye, I honestly, it was a several person process every day to get those drops in. So
kids is a special situation trying to get drops into the eyes of children.
Absolutely. But no, I think that's such an interesting tip too, because I often, wonder as well if someone is experiencing vision loss as well, how you aim it and make sure that you're getting it in the eye. So I think that's a really, really interesting technique to get it going. Another question that we hear a lot is about, you've talked about these drops having different frequencies. So if someone misses a dose or two, is that a significant problem, or is it okay if a dose gets missed?
So patients often ask me this question from time to time. They'll say, hey, I forgot one of my drops last week and 2 drops the week before. Is that a problem? And if you're being consistent with your drops the majority of the time, then overall you're going to be okay. It's the folks who end up missing more than just the occasional drop where things start to become a bit of a problem. And this does come back to what I described earlier in terms of long-acting and short-acting drops. A long-acting drop, once you've used it for a few weeks, it does build up a certain concentration within your eye. And so you have a little bit of a buffer in terms of if you miss one dose, it's probably not the end of the world. But you don't want to be missing too much more frequently than that, because it's got that level there, and it'll take a bit of time before it starts to peter out. Whereas the short-acting drops, because they don't last as long as they sort of fade away a little bit more quickly, you do want to be a little bit more diligent with those, because if you miss a dose here and there, you're more likely to have that overall level of medication drop off much more quickly. So in those cases, I do recommend that people try to be a little bit more consistent with them. And in the ideal world, you'd use your drop every single time that it's scheduled.
So if you're prescribed a particular eye drop and you're on that for a very long time, do these medications lose effectiveness if used for over years, for example? Is there ever the need to sort of switch things up because one of the medicines has lost sort of its effectiveness in the eye?
So I would say it's uncommon for that to happen. And for the majority of medications that we use, we don't typically see that type of loss of efficacy per se. One of the classes of medications, actually, funnily enough, the beta blockers, once again, do have a sort of classically described syndrome where over a long period of time, the effect kind of wears off. And we think that it may have to do with something at the cell level about the different receptors that the medicine talks to and tells the cell to help bring the eye pressure down overall. So sometimes that does happen. More commonly, what ends up happening is that over time, people either their glaucoma itself starts to get worse. So it looks like the medication is less effective, but in actuality, the medication is just as effective. It's just the glaucoma, the disease is getting worse.
So we talked a lot about drops. So the drops that are available now, are there, you mentioned in the beginning, there are some drops that may have other effects as well. Is there any research happening now into developing new kinds of drops or more effective drops?
So we actually are living in quite an exciting little time right now in the field of drops, because for the last 20 years or so, the number of new types of drops entering the market was really quite limited. We had this new set of drops that sort of came into being in the early 2000s or thereabouts, and those kind of changed the face of glaucoma because they were so effective and they had very, very few side effects, and they're pretty much what almost every single glaucoma patient will be on, either at the beginning or at some point of their treatment. But then there was a long period of time when there were people trying to develop drops, but they weren't really going all that far. Whereas in the last couple of years, we've seen quite a few new medications enter the market. And definitely there are lots of studies ongoing right now looking into new types of medication and also different ways of delivering the medication. So not just in eye drop form, because we know eye drops can be a little bit of a pain to use every single day, multiple times a day. And so there is a lot of research into how can we deliver medication in a way where people don't have to worry about putting drops in every single day, multiple times a day. So for instance, there are certain implants that can be physically injected into the eye, where the medication slowly releases over a long period of time. And so that's just one example of a number of different newer avenues that are either out there right now or will soon be out there.
So a question I get often through our health information service too is for people who have multiple conditions. So for someone, for example, who has AMD and glaucoma and is concerned about how drops or getting injections in the eye interact, are there things that people should be wary of if they have both conditions?
Mm-hmm. So, and then just for the audience, AMD is age-related macular degeneration, which is, as the name says, it's on the bottle, age-related, wear and tear at the back of the eye, and that actually damages the central vision of the eye as opposed to glaucoma, which damages typically the peripheral vision. And for certain types of macular degeneration, people do need frequent injections of special medications into the eye to help keep the macular degeneration under control. So in terms of what people need to be looking out for if they do have both, so it's actually an interesting one because it's more something that their eye doctors need to be looking out for as compared to the individual themselves. Because one of the things that can happen for people with glaucoma who are getting injections for their macular degeneration is we know that eye pressure will go up very briefly after the injection is done, even though it's a tiny, tiny volume of medication that's injected in the eye. Eyes themselves are not very big. And so even a tiny injection can lead to the pressure going up a little bit. And so sometimes the retina specialist needs to perform some additional procedures at the time of the injection to help maintain the pressure to stop it from going too high. And the retina specialist probably should be checking the eye pressure from time to time as well, because they'll be seeing the retina specialist much more frequently than they will be seeing the glaucoma specialist, just to make sure that the pressure is not slowly creeping up over time as they get injection upon injection. It's not common at all that that happens, but once in a while it can.
Thank you. And so with eye drops too, and you mentioned something earlier about artificial lubricant, I guess, or artificial tears. So I often get the question about glaucoma. Do the eye drops that you use for glaucoma also, can they help for things like dry eye or eye strain, or are those a different type of drops?
Mm-hmm. So very different types of drops, and funnily enough, glaucoma drops can often make pre-existing dry eyes a little bit worse. And so because of the medications that we use, they can cause some irritation to the surface of the eye, which people can find uncomfortable from time to time. And so it's not uncommon that people who are on a number of different bottles for their glaucoma end up needing to use some artificial tears as well to help with the comfort of the eye. And that's a pretty common thing. What I always recommend to my patients is if they're using their artificial tears at the same time as the glaucoma drops, just to make sure they're waiting about 5 minutes in between the drops, because you don't want one of the drops to wash out the other.
That makes total sense. So this is great. So we've talked a lot about eye drops, and I'm just wondering if you could touch briefly as well on any other treatment options that there are for glaucoma in addition to drops.
Mm-hmm. Yeah, so there's the good thing with glaucoma is that we do have quite a few different treatment modalities. So drops are probably one of the more common ones that we use, and especially in people who are first diagnosed with glaucoma and just have a very early or a mild glaucoma. Drops can be a very reasonable way of keeping the eye pressure under control. One of the other very popular types of treatment options, one that I use in my office very frequently, is what I will describe as a gentle laser. So it's a particular type of treatment where we use a laser or a type of light to stimulate the drainage system within the eye. So this is the natural drainage system within the eye. And by stimulating it through a very interesting biochemical process, we actually are able to get it to become a little bit more efficient. And so by having the drainage system more efficient, that actually helps lower the eye pressure over time. The great thing about this treatment is that once the treatment is performed, you don't necessarily need to be using a particular eye drop afterwards, and that can work for months, sometimes even longer than that. Now, that being said, there are people who are on a number of different medications. So perhaps they're on 2 bottles for their glaucoma, and their doctor says to them, you know what, we probably should be doing a laser to help bring the pressure down. Every once in a while, someone will come back to me and I'll look at them and say, oh no, your pressure's gone up again, even though we just did the laser. What happened? And then we realized that they misunderstood and they stopped all of their eye drops after we did the laser, even though we had the recommendation was to continue the eye drops and that the laser was an adjunct. So the patient just needs to be very clear with their treating doctor as to what the role of the laser is. Is it a replacement for an eye drop, or is it simply just in addition to the existing treatment? Because we use them in both roles very, very frequently. And then beyond the laser treatment, beyond the eye drops, there are also many, many different types of surgeries to help bring down eye pressure. I won't go into all of the details for them because there are so many of them, but those usually we reserve those for after the drops of the laser are maybe not as effective as they ought to be.
So is it the case then that once someone has surgery, will they then not have to use drops after surgery, or is there often there's still drops after the surgery as well?
So I'm going to give you the very, very typical doctor answer here and say that it depends. So occasionally we can get people off their eye drops for their glaucoma, but not always, and sometimes not off all of the drops. One thing for everyone to understand and to be clear about is that the eye drops, the laser, the surgery, what we are treating with that is lowering the eye pressure, the intraocular pressure. But as Dr. Chauhan said at the beginning of the session, that is not the definition of glaucoma. Glaucoma is damage to that particular cell in the eye, the retinal ganglion cell that makes up the optic nerve. So even though we're treating the pressure, which is the risk factor, the thing that we know by bringing it down, we can slow down the glaucoma. We're not curing the glaucoma, unfortunately. So by having the surgery, unfortunately, you're not being cured of the glaucoma. We're just finding different ways to slow it down. So over time, people may still need to be on eye drops.
Okay, well, thank you very much. I think that was all for my questions, and I see we have a bunch of questions coming into the Q&A, so I'll give you just a moment of a breather, and then we'll look at some of those questions from our audience. So in the meantime, I'm just going to introduce one of my colleagues here just for a moment. I can get Jamie here. We'll just bring Jamie in for a moment. Oh, there she is. So I'm just going to turn it over to Jamie for a moment just to tell our audience a little bit more about how they can get involved with Fighting Blindness Canada, and then we'll be back for our Q&A.
Oh, that's fantastic. Thank you, Morgan. Hello, everybody. Thank you all for joining us today. My name is Jamie Alexanderson, and I am the Senior Manager of Development at Fighting Blindness Canada. This has been a great webinar so far. Thank you so much to our speakers. I actually have a condition that causes pressure in my eyes, so I'm grateful to always learn from you, and thank you to the work that you're doing. It's extremely valuable, and I know important to so many, including many of our guests listening to the webinar today, so thank you. I do want to take a few moments to speak with all of you here today, to everyone listening at this webinar, to talk a little bit about how maybe you'd like to consider getting more involved with Fighting Blindness Canada to support vision research. As you know, many of you know, our longstanding supporters out there, Fighting Blindness Canada's mission is to invest in groundbreaking vision research, some of which you're learning about today, and of course wouldn't be possible without our community of donor support. So here I wanted to share with you just a few ways you might consider getting more involved with the organization, with Fighting Blindness Canada. A great way to get involved is our events, which are coming up soon. We have some wonderful events coming up. We have our Cycle for Sight this summer. We have Comic Vision coming back, and of course Ride for Sight. And so please visit our website and learn about our events and how you can get involved, whether you're participating or volunteering. And then another wonderful way that you might consider getting involved is marking a milestone or celebrating a special occasion in your life. You know, perhaps consider making a gift in honor of a friend or a loved one or a colleague. We have lovely cards on our website you can choose from, both by mail or electronic. And if you make a donation, you could learn more about those ways to give in honor of someone that you care about. Also, a big part of what we do at Fighting Blindness Canada focuses on advocacy and public awareness. So making your voice heard on issues that matter to you are really important. So please reach out and share your stories with us. And you know, just to sort of bring in a few more opportunities for you to get more involved and to give back if that's how you wish to make your mark, and you know, a way is to support vision research through we have an employee matching program. So, you know, many people don't know that employers do match donations. A lot of their employees, you know, have donations of charities of your choice. And also other ways to get involved, you can visit our website. If you click on our donate button, you'll be able to see a bunch of opportunities for you to make a donation in various ways that are meaningful to you. So please reach out to us anytime. I know Morgan, our host, will be sending out an email after this webinar with our contact information, and we look forward to hearing from you. So thank you for your time and enjoy the rest of the webinar. Bye for now.
Wonderful. Thank you so much, Jamie. Yes, it's a very exciting time going back to our in-person events and Cycle for Sight coming up this June. Okay, so let's get started on some of your fantastic questions that are coming in here. So the first question we had in here was from Mervyn, and Mervyn's asking, I think going back to something that Dr. Chauhan said, was talking about how you know, the pressure is not always necessarily an indicator. So how can glaucoma be diagnosed if your eye pressure is in the normal range?
Right, that's a good question. And you know, as both of us have said, that the intraocular pressure is not something that's used for diagnosis. It's used for monitoring your therapy. So typically what would happen is that during a routine eye examination, the eye doctor would have a look inside your eye and perhaps also do a visual field test. And if there is something suspicious there, then you would have further investigations done. And so really the diagnosis of glaucoma is made on the appearance of the optic nerve that we've talked about throughout this afternoon, and also the field of vision, which is measured with the perimeter. So those are routine tests that are done, and we have sophisticated ways of doing it. We have some very exciting imaging tools, as we talked about before, and of course measuring your visual field sensitivity that those of you in the audience who have glaucoma know very well. So the diagnosis is made on the basis of that. And then once your eye doctor decides that you need to be treated, the pressure in the eye is of course important because the efficacy of the treatment depends on how much you reduce the eye pressure. And in most cases, that reduction in the eye pressure will have an impact on how quickly the disease progresses. So just to clarify, we need to take this, we need to sort of decouple high pressure and glaucoma, because that's what the sort of lay literature and the lay public always has associated from everything that we read, that when you say that you have a high eye pressure, you have glaucoma, and conversely, if you have glaucoma, you have high eye pressure. So we need to decouple these two things. So it's very important to make that distinction.
Yeah, and I imagine it's like, everyone is different as well. So it's important to know what those eye pressures are as a baseline, so then you can know if pressure is going up or down as the treatment takes place as well.
Right.
So we have another question, Dr. Chauhan, for you from Len. And Len is asking, are the effects on the ganglion cells the same for narrow angle glaucoma?
That's a great question again. So what distinguishes narrow angle glaucoma or angle closure glaucoma from the sort of common open angle glaucoma is the level of the eye pressure. So typically in open angle glaucoma, where the angle, so the angle is really the angle through which the fluid in the front of the eye has to drain. So if you imagine the cornea, that's a clear part in the front of the eye. So the angle between the back of the cornea and the iris, which is the colored part of your eye, forms the angle. And of course, if it's very, very narrow, then you could impede the outflow of the fluid that flows in the front of the eye that controls the eye pressure. So typically in narrow angle glaucoma, what happens is that angle, of course, becomes narrow, and sometimes it can close. And when that happens, you get typically a sudden increase in the eye pressure. So that sudden increase in the eye pressure and the level of the eye pressure, which typically is quite high, basically accelerates the loss of the ganglion cell. So the impact of eye pressure on the ganglion cell is no different. If you have a higher pressure or a very high pressure, it's just the rate at which that's going to go down. So typically what would happen is if the pressure is very high, then the rate of ganglion cell loss is going to be much faster. So the impact on the ganglion cell is just the same, it's just the level of the eye pressure. But having said that, again, as we've talked about, is that each eye has its own level of susceptibility, right? So a certain level of eye pressure can be easily tolerated by some people, whereas some individuals cannot tolerate that pressure. But certainly the levels of pressure that we talk about in angle closure glaucoma are much, much higher, and the rate of loss of ganglion cells is much, much quicker in that case.
Thank you. Another question here, what effects do cataracts have on the onset of glaucoma? Maybe I'll defer to Dr. Kanjee on this one.
Sure. So cataracts and glaucoma are one of those topics where there's a lot of different aspects to it. So a cataract itself, so a cataract is basically the clouding of the natural lens inside the eye. This is a normal aging process, happens to absolutely everybody in the world. So every single person in this world will develop cataracts should they live long enough. And at some point, the majority of people will need a cataract surgery to replace that cloudy lens with a clear plastic lens instead. So there are certain types of glaucoma where as the cataract gets larger and larger over time, it can lead to a slow increase in the eye pressure. So in some forms of what we call angle closure glaucoma, as Dr. Chauhan was describing, you can, as the cataract gets larger and larger, more and more of the angle can slowly close over a long period of time. And that's similar to the process Dr. Chauhan just described, where the drainage system closes quite rapidly and leads to a very sudden elevation in eye pressure, but this is just over a long period of time as opposed to over hours or days. And so in those situations, as the cataract gets bigger, the eye pressure can go up. And there are some very rare situations where a cataract can cause a type of inflammation or swelling in the front of the eye that can also cause the eye pressure to go up. And so that's sort of one of the ways that it can interact. Much more commonly, though, it has really no significant impact on the glaucoma itself. It's sort of a separate process altogether. Occasionally, when we take a cataract out of someone's eye and replace it with a clear plastic lens, we note that the eye pressure goes down a little bit. And that's actually more common in certain populations, in people who have a particular structure to their eye more than others. So the sense that we are having as we see more and more research on this topic being done is that people where they had a tendency for the drainage system to be perhaps a little bit more narrow, so a little bit more along that narrow angled spectrum, may actually benefit a little bit from having their cataract taken out in terms of helping with the eye pressure, as opposed to people who maybe fall on the more typical open angle spectrum. Although even there, from time to time, we'll see eye pressure being reduced just by having the cataract taken out.
Thank you. So Sandra's asking about contact lenses, and she says, should people stop wearing contact lenses? And I'm not sure if she means in general or if you have glaucoma, but maybe could you comment on both?
Sure. So contact lenses and glaucoma, it's fine. You don't have to stop wearing contact lenses if you're taking eye drops. It does sort of speak to a slightly different question, which I'll put my public health hat on for a moment here and just do my usual public services announcement when it comes to contact lenses. So contact lenses are extremely convenient. People really, really enjoy them, not having to wear glasses, and I think that they can be a really great tool, provided that they are being used appropriately. So just people need to understand that a contact lens is a piece of plastic that's sitting on the front of the eye, and it is reducing the amount of oxygen that gets into the front of the eye, because your cornea is the clear structure of the eye that Dr. Chauhan mentioned earlier. Does not normally have any blood vessels in it, so there's no real way for the cells in it to get the oxygen that it needs. So typically those cells actually just take the oxygen in from around us. So if you have a contact lens on the surface of the eye, you're reducing how much oxygen. It's not zero, because the contact lens is designed to allow some, but not as much. And so if you use it many, many hours all day long, every single day, you're sleeping in your contact lenses all the time, you're going to end up with some really, really major problems. So my recommendation is just talk to the person who's prescribing your contact lenses to make sure that you're using the one that is the one that they recommend, and that you're not using them too many hours in a day or too many days a week, or that you're not doing any of the other things that can cause problems. So I know that's not directly related to glaucoma, but someone asks me about contact lenses, I'm going to give my public service announcement.
That's good. That's very important. I think I would not have thought of that. I don't know why. I would think that I have a pretty high prescription. I've never been able to get contacts that work for me, and I never would have thought that. But I guess, yeah, the amount of oxygen that get in is obviously being blocked by that lens. So that's really interesting. So Sandra here has a question about, she says, what about developing glaucoma with work that requires a lot of screen time? She says she used Theraspex, but not all the time. I'm not familiar. I imagine Theraspex is maybe like a blue light blocker or something like that. Is that what Theraspex is?
I'm not actually familiar with that particular product.
Okay.
I don't know what it is either, I'm sorry to say, but there's no evidence that I'm aware of that doing any screen time and glaucoma are related. If I can just add one thing to Dr. Kanjee, my public service announcement as well for contact lenses, because I'm a contact lens wearer, is under no circumstances swim in a chlorinated pool with your contact lenses without wearing very, very good goggles that can keep the water out, because you can potentially get a very, very nasty disease, a nasty infection from a swimming pool if you swim with your contact lenses. So please be aware of that.
Good to know all the things I'm learning about contact lenses today. So Sylvia has a question, and I'm not sure, I'll kind of open this up to either Dr. Chauhan or Dr. Kanjee, is asking if there is any research that's specifically looking at CRVOs and glaucoma. Are either of you aware of any research that looks at those two things?
Why don't you go ahead, Dr. Kanjee?
Sure. So a CRVO is a central retinal vein occlusion. So there's different types of blood vessels that go into the eye. As with any other part of the body, there's arteries that bring the blood in and veins that take the blood away. And for some people, the vein can get blocked for one reason or another. And so that's what we call an occlusion. And when that vein gets blocked, you can get all sorts of different problems in the eye. You can get bleeding in parts of the eye where it should not be. You can get swelling in different tissues, and you can get damage to the vision as well. So there's a million different reasons why people get central retinal vein occlusions or any other type of retinal vein occlusion. One of the rare causes of retinal vein occlusion is very, very high pressure in the eye. So this is sort of the very classically described association that we've known for decades and decades and decades. And central retinal vein occlusions themselves can actually predispose individuals to develop a very particular type of glaucoma, where blood vessels start growing in the front part of the eye where they should not be, and they actually block up the drainage system. So that's sort of the very classic, typical description that yes, there is an association between them. It's not a one-to-one thing. So not every single person who has glaucoma will have a retinal vein occlusion, and not every single person who has a retinal vein occlusion will develop glaucoma, but there is sort of a little bit of a link. And I do believe that there has been some recent research looking into some of the more fine linkages between those two things. I'm not sure, Dr. Chauhan, do you have any comments on that?
No, I think you've answered that very succinctly. There is evidence of clearly an evidence of an association between CRVO and glaucoma, but it's a sort of chicken and egg situation as well, because you'd have to follow so many individuals over time to be able to determine actually how much risk there is. But I think most of the studies that I'm aware of have shown that there is a link between certain types of glaucoma and CRVO, for sure.
I have another question for you, Dr. Chauhan. So will your research that you're working on now, will that help other diseases that impact optic nerve, for example, optic neuritis or LHON?
Yeah, so basically, our research should be applicable to anything that leads to loss of ganglion cells. So basically, any of the optic atrophies, basically that will kill retinal ganglion cells. So you have the ischemic optic neuropathies, you have a whole bunch of other diseases that do lead to ganglion cell loss, actually even diabetic retinopathy. So, our imaging technique should be amenable to all of that. We use glaucoma because we know that's the disease that we know very well, and we know how to create it in mice. So that's what we're working with. But you're absolutely right. These are tools, hopefully, that can be applicable to other diseases as well, for sure.
Thank you. Okay, another question here in the chat from Michelle, and Michelle is bringing up something I think is really important. So she says, how can you talk to friends and family about the importance of taking their drops regularly? She's a friend who stopped taking their drops because he moved to a new city and he didn't have a new eye doctor, and his vision suffered as a result, resulting in blindness in one of his eyes. So do you have tips, Dr. Kanjee, for how to talk to patients about how important it is to take their drops, or how family members and friends can encourage their loved ones to continue with the treatment?
Yeah, this is unfortunately a challenging situation that people can find themselves in. And just in general, it's one of the things that we as glaucoma specialists and any eye physician spends a lot of time counseling patients about, of just the importance of taking their eye drops. For whatever reason, some people do have the sense that unlike medications used for other parts of the body, so for instance, if you had high blood pressure or diabetes, it's well known in just public discourse that people will be on medication for that, and that's just it's accepted in society, and people will be on that medication for a very, very long time. But because something like glaucoma is perhaps not as well known for the average person walking on the street, they may not have a family member who has it or know any friends who have it, and because it can be a completely silent disease where it happens and you don't even know that it's there. I do wonder whether that has influenced the fact that people don't always understand that in many, many situations, once you're on eye drops for glaucoma, it's treatment for life. The glaucoma doesn't go away. You do need to be on those medications. And so what we'll often have the discussion with individuals is just helping them understand that, we do need this medication to be going because we're helping to bring the pressure down. If you take the medication away, the pressure goes back to where it was at the beginning. And certainly involving friends and family in that discussion can be a very powerful tool because they're the ones who know the individual better than we do. They're the ones who spend more time with them than we do. And if they're there to help support the individual and really empower them to keep using their eye drops every single day, then that's going to be a win for everyone involved.
Absolutely, thank you. Helen has a question here. So she said she's been told by her eye doctor that her optic nerve is larger than the average person's, but that it's not enlarged because of glaucoma or any other problem. So she's just saying, so would my doctor have determined that I don't currently have glaucoma because there's not any damage to her retinal ganglion cells?
Shall I answer this, Dr. Kanjee?
Yes, go ahead.
The optic nerve is basically, I talked about earlier on how these retinal ganglion cells, you can imagine them as little cells that have these wires that leave the eye and they go to the brain. So they have to leave the eye, and therefore the eye has to have a hole through which these cells have to pass through. And this is precisely what the optic nerve head really is. It's really the opening at the back of the eye that allows these cables, if you like, to pass through. Now, for whatever reason we don't really understand, some of these holes, some of these optic nerve heads are actually larger than others. In fact, there's up to a 10-fold variation in the area of the optic nerve head. And sometimes there's some evidence that suggests the bigger the hole is, the more ganglion cells you have, the more wires you have to put through that. But that relationship isn't as clear. So basically, that hole essentially has to be closed. You can't just have an hole because you have to have the eye pressure to be maintained within the eye, otherwise the eye would collapse and so on. So what happens is that there's other tissue in addition to these, what we call axons, which are the wires of the ganglion cells, to be able to actually support that hole. So your caller is correct in saying that her eye doctor has said that her optic nerve is large. It's not enlarged. That there's a big difference here, okay? Because she's probably born with this. In fact, almost certainly born with this. And some people, as I said, have optic nerves that are 10 times bigger than others, and we just don't know why. Okay, it's just an anatomical variation. Now, we have tools to be able to detect damage in very, very small optic nerves and very, very large optic nerves. And one of the things about glaucoma is that glaucoma is a progressive disease. So it's very important that if your eye doctor has said, look, I suspect that, you are at risk of glaucoma, and I'd like to see you, in a year's time to just see how things are going, please do go ahead and do that, even if you don't have glaucoma at that particular point in time. Because if your eye doctor can measure a change in your eye, irrespective of whether your optic nerve head is big or whether it's small, that in itself is a diagnostic clue. Okay, so often a diagnosis can be made on the basis of a single examination, but sometimes it requires multiple examinations and evidence that something has changed. And this change can occur whether your optic nerve is large or whether it's small. And your doctor should have the tools to be able to do that. So having a large optic nerve is no concern, really. There's very little evidence that suggests that large optic nerves are in more danger of getting glaucoma as opposed to small ones. There simply isn't that evidence out there, but it's important to watch for change. So that's important to realize. And it's not enlarged. Okay? It hasn't gotten bigger. It's just congenitally like way.
Yeah, that's, I think, such an important distinction because, as you said, enlarge implies that there's been a change, that there is something going on there, whereas it's different from just being born with it, and that's just the variation in your eye. So another question here, is the optic nerve the only cell type or part of the eye that's damaged in glaucoma, or does the high pressure cause other changes or damage within the eye?
What a great question. That's a question that's probably coming from a scientist as opposed to somebody who's a patient. So if the pressure is very high, then it's possible that you can get damage to other cell types as well in the retina. But for the normal levels of pressure, by normal, I use that term very loosely because, there's a range of pressures over which open angle glaucoma occurs. At the moment, the consensus appears to be that the damage is limited to the ganglion cells and to the connections that the ganglion cells make, right? So the ganglion cells make connections also to a type of cell that resides slightly outer to the ganglion cell. It's the middle neural layer in the eye, in the retina, and that layer where they make the connections is also lost in glaucoma. But at this point in time, there's little evidence that other cell types are damaged. Now, having said that, I can't answer that definitively because within the optic nerve head, there are a whole bunch of supporting cells that support these axons as they leave the eye. So there is some evidence that there are changes that are going on within the cells. Whether they are just changes or whether those cells are also being lost, we don't know. But as far as neurons are concerned, these are the special types of brain cells, if you like. So far, the evidence appears to be that it's limited to retinal ganglion cells.
Great, thank you. Our next question is from Jerry, and Jerry is asking, can the pressure in the eye cause any pain? Probably a good one for you, Dr. Kanjee. Yeah.
Yes, so pressure and pain. So it's one of those situations where, again, it kind of depends. Typically, the types of eye pressures that we see day to day are not going to be to a level that are going to cause any pain or discomfort in the eye. People will often say to me, oh, I feel a pressure in my eye, and it's very rare that they're actually feeling their normal eye pressure. The only time that you can actually have pressure in the eye causing significant pain is when it gets to 4, 5, 6 times the normal value, but also when it does that in a very short space of time. So when occasionally people will come to see me and they'll have been referred and their pressures are incredibly high in their eyes, but they've gone up very, very slowly over years and years and years. So going from a number maybe in the teens to a number like 40s or 50s or even 60s occasionally. And so those types of numbers, if they go up very, very slowly, may not actually cause any pain. But if you go from a number of, say, 15 or 20 all the way up to 40 or 60 in the course of a couple of hours, then you're going to have a lot of pain. And typically that will manifest as pain in that affected eye. You'll have some redness to the eye. The vision may become very blurry as other structures in the eye become inflamed and cloudy. You may start seeing halos around lights, again, because of the different cloudiness that happens in the eye. And people can sometimes get nausea and start throwing up because they're having so much pain in the eye. That's very uncommon, but those would be signs of what we would call an acute or a sudden glaucoma.
And if someone did experience that, is that considered like an emergency? Would someone need to get treatment right away for that?
Yeah, for sure. When the pressure goes up by that much that quickly, really the treatment is to get it back down just as quickly, because the longer that it stays at those levels, and when it's gone up in such a short period of time, that can really lead to very accelerated damage to the nerve. So those are the types of situations where someone does need to get themselves looked at very, very quickly.
Absolutely. So I have a question here from Michael saying, can you comment on the relevance of pigments seen in association with a diagnosis of open angle glaucoma?
Sure. So there's different types of pigments that we will sometimes discuss as glaucoma specialists or as eye doctors when we're talking about glaucoma. So often when we're thinking about glaucoma, we think about, is there an underlying reason within the eye that we can see that explains why someone may have nerve damage to their nerve? And the most common explanation that we end up coming to is, well, we don't see any specific thing in the eye that's causing it. And so that is what we often describe as primary open-angle glaucoma, and that would be by far the most common type of glaucoma that we see and treat. And this is as opposed to other types of glaucomas where we say, oh, you know what, we do see another process happening in the eye, and that is what is predisposing to the glaucoma. And there is a number of these where we sort of have this broad umbrella term where we say, you know, there's different pigment that we see in the eye that gives us the clue that this might be one of these other types of glaucoma. And the reason that that can sometimes be relevant is that the way they behave or the way that they're treated might be a little bit different to your more typical glaucoma. So two of the common ones that we see are one that's called exfoliation syndrome or exfoliation glaucoma, and the other one is pigment dispersion glaucoma. And both of these involve certain pigments that we see, or little spots on the eye that the individual themselves will not be able to see. Even if they look really, really hard at their eye, they're probably not going to be able to see it because it's microscopic. You need a very strong microscope to be able to see any of these things. And so when we see that, it's really us saying to ourselves, okay, maybe this is a slightly different type of glaucoma. The way that it needs to be followed might be a little bit different. The treatments might be a little bit different, and some of the considerations that we have might be a little bit different to someone who has a more run-of-the-mill primary open-angle glaucoma.
Thank you. I see two more questions in the Q&A. So if anyone has any questions that they haven't asked yet, this is your last call to get those in for today. So Judith says, not really a question, but she says, I cannot tolerate preservative in any eye drop. Dr. Kanjee, is that a common response from people? What sort of alternatives are available if people have trouble tolerating the drops?
So some people certainly do have issues with some of the preservatives. So typically for an eye drop, you've got multiple different components in it. You've got the actual medication itself that's doing the work, that's actually getting the eye pressure down, but then you need something to maintain the structure and integrity of the molecules within that medication, and you need something to be able to actually deliver the medication to your eye and then keep that medication stable over a long period of time while it's in a bottle sitting on a shelf somewhere. And so often we end up having to put preservatives in these eye drops just to keep them essentially clean. Because if you didn't have them, then there's a possibility that little bugs that are in the air around you could end up getting into it, and you could get bacteria or viruses and things like that growing inside the bottle, which would not be great. So preservatives are there, they're there for a very, very good reason. However, some people do find that over time, having used drops for years and years and years, and sometimes even shorter periods than that, that they do develop irritation or sensitivities to these preservatives or to other compounds within the drops. And in those situations, your doctor may switch you to a different eye drop that has a different type of preservative. They may offer to put you on eye drops that do not have any preservatives at all. So you may end up getting, instead of a little bottle of your eye drops, they may come in these tiny little vials where you break off the cap and there's 1 or 2 drops in there. You use that and then the vial is empty. So you don't need any preservative in there because it's completely sterile until the cap is broken. It's used right away and then it is discarded. Or in some situations, you might be offered a laser treatment like the one that I spoke about earlier, or occasionally surgery instead if no drops are a possibility, but the pressure still needs to come down.
Thank you. That's really interesting about the sort of tiny capsules. It's really interesting to not have the preservatives if you just have a single dose. So we have someone else who said that their husband had to have a pump installed, calling a Bearvelt BG 101 350 glaucoma implant. Now, is that something similar? I know earlier you talked about some treatments that almost like a little reservoir that go in the eye that would slowly disperse medication. Is that what that is?
Yeah, so the Bearvelt is a type of glaucoma surgery that we do. So it falls into the category of what we describe as glaucoma implants or tubes that are placed in the eye. And these are used to help drain fluid out of the eye, to help bring the eye pressure down. And there's many different indications for them. Sometimes they're used as an initial surgery. Sometimes they're used after other glaucoma surgeries have stopped working. And the Bearvelt tube is a particular type within that category. It's not like the technology I was describing earlier, where a medication is injected into the eye and slowly releases medication over time. This is really just a conduit, an alternative pathway to allow fluid to drain out of the eye in those situations where we feel the natural drainage systems in the eye are simply no longer functioning and cannot be restored. And so we need to create a new drainage system instead, in which case we may do a surgery such as implanting a tube into the eye.
Thank you. Question here from Pamela to Dr. Chauhan. Are different types of ganglion cells affected differently in different types of glaucoma?
Yeah, again, this is a question probably from a scientist who's asking a question like this. So that's a great question. So we have so many different subtypes of retinal ganglion cells. Some ganglion cells respond preferentially to moving objects, for example, so things that move fast in your peripheral vision, and other ganglion cells are really concerned with color and form, so how fine print you can read, for example, and so on. And they're not too interested in motion or moving targets. Some are interested in contrast and so on. So there are a variety of different types of cells that exist, of ganglion cells that exist. Now, the evidence that a specific subtype of ganglion cells is preferentially lost in glaucoma is very controversial. It's been controversial for the last 3 or 4 decades because some scientists say there is and others say that there isn't. The reality is that all ganglion cell types are damaged in glaucoma. It's possible that some are more preferentially damaged, but ganglion cell loss is not exclusive to a particular subtype. So it's not like saying, oh, only the cells that take care of moving objects, for example, are damaged, whereas those that are responsible for seeing color are protected. That's not the case. So it's not a cut and dry like that. All ganglion cell subtypes are damaged. It's possible that some are more susceptible, but we still don't know the definitive answer of that. So it's a good question. I wish I had a clearer answer for you.
Still work to be done, right? There's always
more work to be done, exactly.
I love our audience. They always come in with a final burst of questions here. So we have another one here from Mervyn who's saying he's confused as to the relationship between eye pressure and damage to the optic nerve. Yeah. If your pressure is in the normal range, but you have damage to the optic nerve, how will reducing the pressure help? And similarly, how does that optic nerve become damaged if the pressure is normal?
Great, that's a great question, and I clearly didn't do a good enough job, so I'm going to try again. So let me explain a little experiment to you. Let's suppose that you took 10,000 people in Halifax, where I live, and you measured their eye pressure. So what you would do is you would see the range of eye pressure in this population. You would see what the average is, and you would see what what the range will be and how many people have certain levels of eye pressure. And then you could say, well, I'm going to define normal eye pressure being as somewhere where 95% of those eye pressures lie. Okay, so that's in statistics how you do it. So when you say something is not normal, it typically applies to that 5% who doesn't fall into that. So if you take that top 5% of people who have the high eye pressure, and you say, okay, now I've identified these individuals, and I'm going to see how many of those people actually have glaucoma. Right, so you do your visual field test, you do your optic nerve imaging and so on, and you find that lo and behold, the vast majority of those people who have statistically high pressures actually don't have glaucoma. And if you follow them over a period of time, actually the vast majority of them will not develop glaucoma. Now, let's take every individual, all of these 10,000 people in Halifax, and you give everybody a visual field test, and you give everybody an OCT test, and you do all of these things, but you don't measure their eye pressures, and you say that my diagnosis of glaucoma is based on what Dr. Kanjee said and what I've said. What you're going to find is that half of the people who have glaucoma in that group actually have statistically normal pressures. Right, so there's the decoupling between pressure and glaucoma. Now, having said all of that, that doesn't necessarily mean that a pressure which is statistically normal is not damaging to a specific eye, right? So a pressure that could be average could actually be highly toxic for a particular individual, right? And therefore, reducing the pressure, no matter whether it's statistically normal or whether it's statistically elevated, will actually have a beneficial effect in trying to slow down that disease. So I hope that answers the question, because we need to decouple glaucoma from pressure, but pressure, no matter at what level, can damage the optic nerve head.
I think that's a fantastic explanation. Thank you. That's, I think, such an important note. And I think that's the note we're going to end on today, because we are out of time. So thank you so much, both to Dr. Chauhan and Dr. Kanjee, for being here and for being generous with their time. It's been really interesting. I've learned a lot. So thank you both for being here. Really appreciate it.